Patrick, thank you very much for your kind words and thank you for the kind invitation. I regret that I cannot attend in person. I would like to transport myself to the city of light and to imagine myself in the amphitheater blue, but unfortunately this year it can't be done. Nonetheless, the fact that these are pre-recorded thanks and a pre-recorded talk does not diminish my sincerity for your kind words and for the acknowledgement. You have asked me to talk on hepatitis B cure, how and when. This is a challenging talk, and it is somewhat presumptuous because I don't know how, and I do not know when we can cure hepatitis B. But we all know that data is emerging, proof of principles have been established, and the needle is clearly moving in a very competitive landscape. We know a great deal about the hepatitis B replication cycle, and this promises many new druggable targets. The replication cycle has been identified. We can identify entry, uncoating, import into the nucleus, development of the cccDNA, transcription, encapsulation of pregenomic RNA, minor strand and plus strand synthesis, virus particle assembly. All these are amenable to inhibition. Nucleoside analogues do not reduce the cccDNA, but new capsid assembly modulators are adding an extra choke point to our ability to inhibit the replication cycle, and also new RNA interference strategies are greatly reducing HBsAg in serum to a degree that we never could achieve with nucleoside analogues. So this represents real progress. However, we have to contend with the fact that hepatitis B surface antigen, the hallmark of chronic hepatitis B, is produced in a manner that is replication dependent, depending upon the replication cycle and the production of virions, but also in a manner that is not dependent on hepatitis B replication. Hepatitis B virus genomes will integrate into host DNA. The linear integrations do not circularize to produce pregenomic RNA and are therefore not involved in the replication cycle, but they do produce subgenomic RNAs from which are transcribed and translated a vast excess of small subviral spherical particles, which we think probably have an important role in inhibiting and exhausting the T and B cell response to hepatitis B. We know that the spherical 22 nanometer subviral particles are in fact produced in great excess and in serum far outnumber the number of hepatitis B DNA containing virions. We also know, and these provide convenient new biomarkers, that we can detect hepatitis B RNA within the serum, within hepatitis B capsids. So a great deal has been learned, and we're able to translate some of this into new strategies. However, a functional cure remains a major challenge, even if we just measure functional cure by HBsAg seroclearance alone. Some of the reasons for this are shown on this slide. The replenishment and the stability of the mini chromosome, cccDNA, by measures such as a shuttle amplification pathway, make it very difficult to eradicate cccDNA. cccDNA is not rapidly degraded by the current nucleoside analog therapies we have. HBsAg production, as I point out, can be produced without a replicative cycle. The genomes integrate in a random fashion into the HBV genome. We can identify long reading frames now, which possibly encode pre-S as well as S protein. The random scattering of these integrations throughout the human chromosome means that such integrants will not be amenable to any measure to dissect them out. And importantly, the very high surface antigen load compared to core antigen almost certainly plays an important role in antigen-specific immune dysfunction and T and B cell exhaustion. How are we currently defining cure? Functional cure has been defined as a sustained loss of HBsAg for greater than 6 months after cessation of therapy. It's a measurable endpoint, does have that advantage, and it's a point at which we would consider indeed discontinuing therapy based on previous results with current therapies. We think that a functional cure will require both profound inhibition of the replicative and as well as the non-replicative hepatitis B cycle and probably immunologic control. Will it improve prognosis? We're not yet sure about that because we have not achieved complete eradication from the host. My own bias is that we will have to achieve HBsAg seroclearance at a young age before the onset of cirrhosis for this to be meaningful. Is there a place for the next tier down of a partial functional cure? This has been defined as a decline in HBsAg to lower levels after finite treatment. By definition, these patients remain surface antigen positive, but they are reset to a lower state. How low is a low HBsAg level? Do we need to reach an absolute level, less than 1,000, less than 100, or a log decline to affect prognosis? Will this improve patient prognosis? And will the regulatory authorities accept this point? These are all unanswered questions. Current therapies for cure include nucleoside analogues, pegylated interferon, the combination and the cessation of nucleoside analogues. And I'm not going to dwell on these because it's not the subject of today's talk. Certainly there is much interest in dual therapy, add-on therapy, and switch therapy with nucleoside analogues and PEG interferon. You have contributed to these studies, and this has been reviewed. So I will not spend time reviewing the subject. It is of interest, and it may point to a residual role of PEG interferon as an immunomodulatory therapy in cure strategies. The other point, of course, is cessation of nucleoside analogues. Again, this has been closely looked at. There are two aims to this approach, whereby we stop nucleoside analogues in patients who have been on long-term therapy. The first aim would be to enter an HBsAg positive low replicative state. This is achievable, but there is always the risk of relapse. The second, more intriguing aim is to trigger HBsAg loss, as you know, by stopping the nucleoside analog. The results are highly variable, and off-treatment loss occurs in recent trials in 4 to 10% of patients and may perhaps reach 20% in carefully selected patients. But the range associated with HBsAg loss is very wide, anything from less than 10, less than 100 to 1,000. And there are many unanswered questions as shown on the right. Are there ethnic influences? Will the type of nucleoside analog therapy influence the timing of response? So we need much more precise information before cessation of nucleoside analogues will be considered as a primary mainstream measure for most patients, but it is a consideration. It's not the subject of today, however, and I've tried to simplify this from a clinical standpoint as two main strategies that could be used for a finite functional cure. The first strategy involves a reduction in HBsAg production and expression, leading to ultimate HBsAg seroclearance, and the subtext is that there is immunologic clearance which sustains HBsAg loss despite residual genomes present in the liver. The second strategy for a finite functional cure is to deepen the inhibition of hepatitis B replication. And I'd like to deal with the first strategy first, HBsAg knockdown strategies. We have a number of newer compounds that interfere with the translation of HBsAg or the assembly of HBsAg. These reduce HBsAg apparently directed to both subviral particle morphogenesis as well as to the morphogenesis of complete virions. The mechanisms are varied, but they are able to target HBsAg derived from virion production, as well as HBsAg derived from integrated viral genomes, a hitherto relatively inaccessible source of HBsAg. They include RNA interference, as you heard today, and I won't review this in detail, which interfere with translation and include small interfering RNAs, locked nucleic acids, and antisense oligonucleotides. These result in destruction of HBsAg RNA. There are second-generation antisense oligonucleotides in trial. There are NAPs and STOPs, which are an interesting class of amphipathic phosphorylthioate oligonucleotides that apparently selectively inhibit HBsAg particle assembly and hence secretion, but are not sequence dependent. And there are a number of interesting new entry inhibitors and monoclonal antibodies. There are lots of variations. Again, it won't be my task to review all of the data. I know that this will have been covered in earlier sessions today. But there are a number of new small interfering RNAs and antisense oligonucleotides which are targeted to different trigger locations. They include the S gene, the X gene, or a combination of both. They are interesting. They may explain some of the variation we are seeing, but fortunately, we have now been able to devise small interfering RNAs that target S production from both integrated as well as episomal RNA transcription. There are slight subtle differences between RNA interference and NAPs and STOPs, and I've oversimplified this in this diagram. We know, for example, that RNA interference in theory and the nucleoside polymers, as well as the STOPs, would actually interfere with subgenomic RNA HBsAg production from integrated viral genomes, but would not affect HBsAg production from cccDNA or directly affect cccDNA because of the different dichotomous assembly pathways for the virion compared to the spherical subviral particles. RNA interfering agents, in theory, might be able to target HBsAg derived from both cccDNA as well as integrated viral genomes. What are the prerequisites for HBsAg knockdown, and what are some of the questions? We would know, and I think are beginning to recognize, that HBsAg protein follows different assembly pathways, and the chaperoning of the subviral particles as well as the complete HBV virions is being elucidated, so we can identify the appropriate targets, as will be shown. RNA interference and nucleic acid polymers reduce HBsAg concentrations on treatment, and in some cases, some persistence of that reduction is observed. There is great heterogeneity, and we do not know what explains the range of decline and the heterogeneity of decline. These could be due to host factors or perhaps viral factors. It may be that hepatocyte cytolysis is required to sustain this response, and our proxy measure for that, crudely, is a serum aminotransferase elevation, but we do need much more granular immunologic detail. We don't know whether a rapid precipitous response is required and what additional immunomodulatory strategies are applied in nature and what might be applicable therapeutically. And we need to know a lot more about whether possibly pre-S1 proteins derived from integrated viral genomes might still be having an effect upon the capsid due to the known capsid interactions. There's much work to be done. Fortunately, there is progress in the field, and I've chosen an example from NAPs and probably STOPs to illustrate this. We know that the assembly of the spherical 22 nanometer subviral particles, which constitute more than 99% of circulating HBsAg, is different from the assembly pathway of the HBsAg filaments and the Dane particles. We also know that the protein chaperones, which actually guide HBsAg assembly from the endoplasmic reticulum to the Golgi body, also differ. It has recently been shown with reasonable certainty that NAPs, for example, target a particular chaperone, DNA JB12, but do not inhibit other chaperones involved in the morphogenesis and assembly of the complete virus particles. So that level of granularity should help us to identify appropriate methodologies and strategies and to fully understand mechanisms. I'm now going to turn to the other strategy, having briefly given an overview of the how of HBsAg decline. What about inhibitors of hepatitis B replication? To inhibit replication, shut it down, and perhaps clear virus thus. Well, there's much interest in the capsid assembly modulators. There are two major classes. Class A or class 1 results in aberrant core protein aggregates. Class N, also known as class 2, result in normal but empty capsids. In other words, prevent the encapsulation of pregenomic RNA within the nascent capsid. There are clear effects of the capsid assembly modulators on hepatitis B replication, indicating that a further checkpoint in the replication cycle has been targeted. They deepen inhibition, they interfere with capsid assembly and inhibit pregenomic RNA, and we can measure that with ultrasensitive tests for DNA and usefully sensitive tests for pregenomic RNA in serum. So the capsid assembly modulators, by definition, inhibit a part of the replicative cycle of hepatitis B, and the effects we will therefore see on various markers will differ. We can see, for example, in this study with H0731 plus a nucleoside analog in E-positive hepatitis B, that there is quite a profound effect on pregenomic RNA, as one would expect. The authors of the study have shown, in fact, that HBV DNA is also decreased. There's some loss of E antigen and correlated antigen, all indicating a reduction in transcription from cccDNA. In the E-positive patient, there is some decline in HBsAg in individual patients. In some, it is considerable. In most, it is relatively modest. I won't show the data, but in fact, in the E-negative patients, the decline in HBsAg is really quite modest. It's typically of 0.5 log decline, probably because the capsid assembly modulators are targeting replicative cycle of hepatitis B. And this is the explanation. For HBsAg, we usually see a relatively limited decline when capsid assembly modulators are used. And probably that decline is accounted for by the decline in complete virions and HBsAg associated with the morphogenesis of complete virions. So there's no dominant effect on HBsAg, particularly on the excess of HBsAg derived from the integrated viral genomes. And indeed, there's an even lower reduction of HBsAg in the E-positive nucleoside analog suppressed patients and E-negative patients. We do see remarkable reductions of pregenomic RNA. This is the hallmark of capsid assembly modulated treatment because we are interfering with the incorporation of the pregenomic RNA into the capsid and subsequently replication and virion production. And this, of course, is very significant in E-positive patients. HBV RNA concentrations are already low at baseline in the E-negative cohorts. So we have much to learn regarding the population-dependent reductions of HBsAg from Dane particles versus from subgenomic RNAs. There's some problems with capsid assembly modulators or capsid inhibitors. They clearly require long-term use. We have seen signature mutations. That does not seem to be a problem when they are applied, as they will be, in combination with nucleoside analogues. Some may result in core and capsid intracellular accumulation. Others will result in empty capsid, and this will not apply. And we have to be on our guard for new toxicities with new agents and, of course, drug-drug interactions. So clearly, I've tried to show you for the how of curing hepatitis B, we have knockdown of surface antigen, but that won't affect the replicative cycle of hepatitis B. We can choke off, deepen inhibition of hepatitis B replication, but that in many patients will not have the prerequisite effect on HBsAg. And the answer, therefore, lies in combination. Some of this data will have been reviewed this morning, and it's not my place to review it again. But clearly, RNA interference is being studied in combination with capsid assembly modulators. There are many programs in the offered extended treatment programs are unfolding, and we will have good data in 2021. So to close with immunomodulatory therapy, this in my view is a lost opportunity. It has lagged behind antiviral therapy. We are looking at multiple immunomodulatory agents, including checkpoint inhibitors, therapeutic vaccines, immunologically engineered T cells, all of which may overcome T and B cell exhaustion and activate responses. But our expectations have been set very high. We're also looking at toll-like receptor agonists, which have been intensively studied, but the expectations to date that immunomodulatory therapy would be affected in the setting of very high surface antigen loads and tolerized T cells was always unrealistic. However, we've now embarked on a new era where immunomodulatory therapies are much more likely to be effective and feasible after a reduction in the host antigen burden. And this is the time for immunologists to provide us the appropriate signatures that define hepatitis B in the various phases of the disease, and also to capitalize in the opportunity we present to them of a profound knockdown, 2 to 4 log, very rapidly in HBsAg and the resulting perturbations in the immune response that determine response. And it may even be a time where we can invoke better immunomodulated therapies, but use PEG interferon or thymosin, old compounds, as proof of principle compounds. Such a proof of principle has been determined in the mouse model in data recently published by Michler et al. What they did was to use a transduced mouse model, use small interfering RNA to rapidly knock down HBsAg and follow with a therapeutic vaccine, and show, in fact, in the treated animals that there was indeed clearance of HBsAg. Mice are not men, but this provides an important proof of principle for knocking down HBsAg and controlling hepatitis B replication and possibly invoking an immunomodulatory strategy subsequently. There are other exciting potential strategies that are unfolding. They include CRISPR technologies. It would not be feasible to envisage that we could actually use these to scissor out integrated viral genomes, but we may be able to target and cleave cccDNA or specific genes such as X. Epigenetic silencing won't be able to do this in a blanket fashion because of the risk to the host, but we might be able to, again, target X in particular and to reduce gene transcription and capitalize on the known interaction of X and its modulation of cccDNA stability to promote degradation by knocking out X with gene editing. And there are strategies such as neutralizing monoclonal antibody, surprising perhaps, but early data is encouraging. So finally, and I won't spend a lot of time on this, what about the when? These are EASL guidelines. We recognize that there are a group of individuals with very low levels of replication where disease progression is low and they're not treated, the inactive carriers. We also recognize patients with very high levels of virus, but minimal hepatic inflammation where they're not treated. And we recognize the other phases of E-positive chronic hepatitis B and E-negative where clearly treatment is moderate. We might need horses for courses for these individuals with low levels of replication, but who might have high quantities of HBsAg because of a large number of viral integrants. Knocking down HBsAg, immunologic clearance could be feasible. For these individuals, and most of the other cases, we would want to shut down replication, knock down HBsAg, understand the heterogeneity of the T cell response, and in fact, restore T cell immunity. We're going to need appropriate strategies for all these different phases, but we may well extend treatment. This is a WHO analysis which has recently been produced. It's a systematic review of treatment indications, both in community reports as well as healthcare facility reports, and they suggest approximately 20 to 25% of individuals are currently eligible for treatment. If we had treatments that would knock down HBsAg safely after a short course of treatment, we may well extend treatment to all HBsAg positive patients to reduce stigmatization, to reduce the cost of follow-up, to reduce the costs of maintenance care. That remains the win of the future of hepatitis B cure. So these are my conclusions. Once again, Patrick, thank you for the invitation. I think we've learned a great deal. We've learned a lot about the replicative cycle of hepatitis B. Getting it right will mean that we'll have to apply this knowledge to the right combinations and sequential strategies, define outcomes as treatment evolve, use newer biomarkers, capitalize on HBsAg loss to identify immunologic signatures that prime responses, use different regimens for different hepatitis B stages and phenotype, be careful about evaluating toxicities and interactions, and above all, keep these treatments affordable. Thank you very much. Once again, thank you very much, Patrick, for the opportunity to participate, and enjoy the rest of the meeting. Thank you.